several papers, all of them dealing with one amazing discovery. And it has to do with the genetic code. This is a statement, another statement made by Richard Dawkins in his classic book, The Blind Watchmaker. He wrote, "The genetic code is universal. I regard this as near conclusive proof that all organisms are descended from a single common ancestor." So he's saying that because the information in the genes in your genome is coded in the same way as the genes in the bacteria that live in your gut, this is incredibly strong evidence, proof almost for him, that you and your bacteria share a single common ancestor, as you do with every other living thing. So let's take a look at this claim, because over the years it's become not so impressive. First of all, here's a diagram of the genetic code, and I've just color-coded the various codons, indicating the general chemical class that the amino acids that each of these three-letter words code for. You don't have to understand it in detail if you're not a biologist, but you can probably see that it is not random. It's actually very, very, very designed, and it does all kinds of amazing things because of the way that codon meanings are assigned to specific amino acids. We won't go into that detail at the moment, but it's really worth a deep dive if you're interested in these things. Lack of variation in codon meanings across almost all phyla is taken as an indicator that initial assignment must have occurred early during evolution, and all organisms must have descended from just one individual with the current codon assignments. So what do we do when we see variations in the genetic code? Turns out that there are 33 genetic code variations that are known. 13 in mitochondria, 2 in plastids, those are things like chloroplasts in plants. The rest in the nuclei of mostly single-celled organisms. Note, by the way, that at least one of these codon variations shows up in mitochondria that are found in some other kinds of organisms, as well as the nucleus of some separate kinds of organisms. It's quite remarkable. These variations, they're not necessarily massive, but they are significant, and there's a lot of them. Exceptions include modifications of the stop codons. There are 3 stop codons, UAA, UAG, and UGA. And to me, those are particularly significant. Some people try to dismiss them. When you understand the mechanism by which stop codons work, that is not an insignificant thing to change from meaning stop to meaning something else, a specific amino acid instead. Here are just a few of these changes to give you an idea of the sort of thing that you get. You can see, for example, in tetrahymena, that UAA and UAG, the common meaning in the what's called the universal genetic code, that would be your genetic code, is stop. But in tetrahymena, it is glutamine. And paramecium, you see the same thing. Paramecium, you probably saw those in biology class or something like that. These are quite common organisms. And you can see there are others. There's a kind of yeast, Candida, CUG, which usually codes for serine, codes for leucine instead. So these are not gigantic differences, but they are significant differences. Any organism that suddenly decided to change this meaning would probably die immediately. Changing the meaning of codons, when you look at the mechanism by which all of this is translated in the cell, this is just not something that you would expect to happen. Here I have laid out just some of the changes in meaning as you go up an evolutionary tree, and this is just in some mitochondria in different groups of organisms. And as you can see, you have things going backwards and forwards, and it's all very, very strange indeed. It would mean that AUA changed from isoleucine to methionine, then changed back to isoleucine in the echinoderm mitochondria. AAA must have changed from lysine to asparagine twice. UGA must have changed to tryptophan, then back to stop again. That's a really big change. And differences in mitochondria lower the number of tRNAs needed. That's the general trend that you see there. So there is a reason why these differences exist, but making the change without engineering everything else to go along with that change is something that would presumably be lethal for the organism and certainly not adaptive. So what's going on with the difference that is reported in all of those papers that I showed you at the beginning of this section? These chemicals are what are represented by the A, T's, G's, and C's that you saw in the genetic code. You can see that there are some of them that have 2 bonds between them, and there are some of them that have 3 bonds between them. They are not going to get muddled up typically about which base pairs up with which other base. And this is vital to get accurate copying of our DNA, among other things. Adenine and thymine are the bases that do 2 bonds. There's a hydrogen on adenine that has a slightly positive charge on it. There's an oxygen on thymine that has a slightly negative charge on it, and they hydrogen bond. So there's that. Then there's this positive charge on the hydrogen down here that you can just see there, and there's a negative charge on the nitrogen that's close to it. So there's another hydrogen bond that forms. This allows these 2 chemicals to recognize one another with great specificity. But it was found in a specific kind of virus, of all things, that they use a different chemical. Instead of using adenine, they use aminoadenine in their genetic alphabet. Aminoadenine is symbolized with the letter Z or Z. So how does this work? There's our 2 normal bonds that we have, but look, there's a negative on that oxygen and there's a positive on that hydrogen, so it can form a 3rd bond. So this is a whole different thing. In this particular virus, we get triple bonds between aminoadenine and thymine and guanine and cytosine, as we do in normal DNA, if you want to think of it that way. This is the equivalent of writing my name, Timothy, in a different alphabet. Here I've written my name in Cyrillic to show you how different it is. Yes, there are similarities there, but the difference is really significant. This is just amazing. It's completely out of thin air almost. I don't know anybody who would have predicted another letter of the DNA alphabet. So what are the implications of this? Viruses are technically not living organisms, but they use the systems found in living organisms to reproduce. Using a different alphabet is thought to protect those viruses that use aminoadenine from some defense mechanism found in bacteria. The idea that the universal genetic code supports common ancestry is an overstatement, because the genetic code clearly isn't completely universal. We need to rethink viruses. That's a huge implication of this. The use of a significantly different chemical alphabet by some viruses raises even more questions about how compelling arguments about the genetic code or genetic systems are when based on the idea that they are all the same. It also demonstrates just how surprising nature is, especially biology. I love being a biologist. So what's the bottom line here? For me, it is that the genetic code obviously looks designed, and it requires brilliance and design to make modifications to it while keeping organisms alive. On the other hand, it looks impossible to realistically evolve changes or differences in the genetic code. The fact that we see those changes is even more suggestive to me of some kind of brilliant intervention, design in biology. And of course, when I think about that, I think about the designer who is revealed in Scripture.